this is video 2 of this topic, our topic 15 variation topic, and we're kind of moving into our statistics now. So you need the Student Notes 15A t-test for this topic. Okay, so we're going to be using a little bit of what we learned at the end of the standard deviation, and we're going to be practicing and applying that before we move on to completing t-tests. Don't worry, there's going to be plenty of practice, and again, you don't have to do any of the calculations in your examination. So I'll make sure I'm very clear about what you need to be able to do in the exam, but we do need to do some calculations when we return to college and we do our practicals to show in your lab books that you have had a go at doing calculations for stats tests, although you are allowed to use computers for that. So we can use Excel packages, and that makes it a lot simpler than using the calculator. Okay, so let's get cracking. Okay, to start, I would like you to go to question 8 in your question pack, topic 15 question pack, and I would like you to do that question for me. Okay, so pause now and do that question. Okay, you should be rejoining me now having done question 8. So we're going to review question 8. Okay, right, so question 8A. What is the name of a position of a gene on a chromosome? So you should have locus or loci to get your mark point there. What is meant for B by genetic diversity? So the number of different alleles for all genes in a population is what you should have down. Okay, now it gets a bit more complicated. So I will make sure, so you've got this in the PowerPoint. So if you can't read it on the screen in your video, then don't worry, you can refer to the PowerPoint as well, but I will verbally go through. Okay, so you're given this investigation into genetic diversity between 4 dog breeds. Okay, and the data in the table that you're given is of course mean, and it's a mean of genetic diversity. The closer to 1, the higher the genetic diversity it told you within the stem, and then the standard deviation. Okay, so the first thing that I did was a little bit of adding to and taking away from to make sure that I know which overlap and which ones do not in terms of standard deviation. Okay, so you can see for the Airedale, I've added my standard deviation to and from, kind of noted down the bottom value and the top value for the standard deviation, the spread around that mean. I've done the same for the miniature terrier. So it says, what do these data show about the differences in genetic diversity between breeds of dog? Okay, now as I've said to you, if you're given standard deviation, you are expected to use it. So I've gone first for the Jack Russell. When I looked at the Jack Russell, the standard deviation is tiny. Okay, so it goes from 0.75 to 0.76. It's only got a standard deviation of 0.01. So I didn't even need to really look, write it on anywhere. I was confident I would be able to say it has a significantly greater genetic diversity compared to all other breeds. Okay, but they want me to put evidence to support this. Okay, so I've said as the mean is closest to 1, and the standard deviation does not overlap. Okay, so the standard deviation does not overlap with any of the other breeds' genetic diversity. So that was a good 1 mark. Okay, now I started to run out of room because I've got big handwriting. I'm hoping you run out of room too, because I don't think you could possibly have got enough mark points in the space that you were provided by me. Okay, so maybe I should have put some more lines in. The Bull Terrier, okay, when I looked at the Bull Terrier, again, small standard deviation. So if I take that 0.02 away, we're at 0.36, and then if I add it to, then I'm at 0.02. 4. So again, that didn't overlap with any others. So I can say confidently the Bull Terrier has a significantly smaller genetic diversity than the other breeds. It's got a very low mean and its standard deviation did not overlap with the other breeds. Okay, so what else can I say? Well, when I looked at the Airedale and the Miniature Terrier, they do overlap just. So actually, I've been able to use that to make a statement too. The genetic diversity of the Airedale Terrier and the Miniature Terrier are not significantly different. So they have a similar genetic diversity. The standard deviations overlap. Now that is an example of a full answer. So that's the kind of detail level that the examiner is expecting you to provide, and every time using your standard deviation. Okay, part D. The Miniature Terriers were first bred from Bull Terriers in the 19th century. Now, suggest, remember, is nobody will have taught you the answer to this question. So suggest are, come on, come up with an idea, think around what you actually know and give an explanation. Okay, so you have to go back to the data and go, okay, so they started with Bull Terriers, and actually the Miniature Terriers got a greater genetic diversity. Okay, now 19th century, so if we're thinking about mutations, really that's over a bigger period of time. Okay, so it's not going to, you're not going to get enough mutations to cause that kind of genetic diversity increase in such a short time period. I know it doesn't feel short, but in evolutionary terms this is a very short time period. Okay, and remember they're also being bred, so that gives you a, you know, selected for breeding. So a suggestion might be, well, the first thing is, have you noted that the Miniature Terriers have a greater genetic diversity than the Bull Terriers from which they were bred? Okay, so the Miniature Terriers, I've said, so this breed has got more alleles in its population. That's a measure of genetic diversity. These must have been introduced by doing less inbreeding or selecting a wider range of characteristics to breed for in the population, whilst the opposite's true for Bull Terriers. So by that I mean, in this group that is the Miniature Terrier, the breeders are being less selective, picking a greater range of characteristics of different dogs within that terrier breed to breed on. Whereas in contrast for the Bull Terriers, they are being inbred. So individuals quite similar are being chosen to breed together to keep those very specific characteristics in the offspring. So that one's quite tricky. Don't worry if you've not quite got it yet. We've got much more thinking about this to do. Okay, so in the last video I said I would make it very clear what you need to be able to do and what you don't need to be able to do when it comes to statistical tests. Okay, so you will need to have evidence in your lab book that you've completed different types of stats tests. Okay, so we will do some calculations. We will practice some calculations this week as well, but we won't ever be asked to do that in the exam. Okay, so you will never be asked to calculate a statistical value in your exams. You will never be asked to calculate standard deviation. So if you can't get your head round doing the calculations of the t-tests, there are some shortcuts I can show you in Excel. You can literally use the Excel package to do all our stats tests. So this week when I've asked you to do t-tests, I've given you standard deviations. I'm not expecting you to calculate them because you'll never be asked to in the exam. Okay, and we can do them together in class when we're doing our practical work. But you will need to pick the right test to use, and there are 3 different stats tests that you need to know about for biology. You will need to interpret statistical values. You will have to write null hypotheses, and you will need to draw conclusions for statistical results. Okay, I will change this to it's always on the exam. So those things you do need to be able to do, okay, are always on the exam. So we make sure we prepare for those really well. We'll make sure everybody can do all of these, and we're only starting the process now. For some people it takes a little bit longer to get your head around stats. That's okay. All right, so which stats test? Okay, so as I said, you have got 3 that you need to know about, and this flowchart is kind of what the exam boards say you should use when you're thinking about stats. Okay, so they say when you are looking at your experiment and you're planning your experiment, thinking about what stats test you're going to use at the end of your experiment, you'll decide if you're looking for an association between data or differences between samples. Okay, so an association is a correlation, a relationship. So if you're looking to see if when you change the light intensity, the rate of photosynthesis changes, then you would be looking for an association between one variable and the one you're measuring. So what you change, you're expecting a relationship with what you measure. Okay, so you'd be looking to see, is there a correlation between my 2 variables? And so you would do a correlation coefficient. Okay, but if you weren't looking for an association, a relationship, you would be comparing your data. Okay, now looking for differences between the samples. If the comparison you're making is between expected frequencies and observed frequencies, then you'll do a chi-squared test. Okay, so an example of this is very much within genetics, and we do lots of these in genetics because we understand how a particular gene is inherited. When we cross, say, 2 heterozygous plants, we would expect that the offspring would fall into 2 categories in terms of the phenotype. All the homozygous dominant and heterozygous individuals would have one characteristic, and the homozygous recessive the other. So we expect that in a 3-to-1 ratio, and we could say that we are expecting for every 4 offspring, 3 to be the dominant characteristic and 1 to be the recessive characteristic. So we're looking for an expected compared to observed, the real then when you actually breed the plants. Okay, so we'll use that a lot in genetics. And finally, the one we're going to be looking at today is when you're actually looking to see if there's a significant difference between means. Okay, so between the mean height of one population and the mean height of another, what would we be using? We're going to use a student t-test. Okay, so just to see whether we've got an idea of any of those, we're just going to concentrate on 2. Spearman's rank correlation coefficient. Okay, so that's what that one's called, Spearman's rank correlation coefficient, and the student t-test. Okay, so if you stop the video, can you decide which of those 2 stats tests, you can go back and look at this sheet as well, you would use for each of these? Okay, and then join me back. Okay, so what stats test would you use if you wanted to investigate the effect of salt concentration on the dry mass of a cabbage? Okay, well, you would be looking for a relationship. You're changing one variable and looking at measuring another, and so you'd be looking for a correlation. So it'd be Spearman's rank or correlation coefficient. A for the first one. If you're investigating into the number of insects found at site A and site B on a rocky shore, then you will be comparing two mean numbers, okay, because obviously you would sample at many sites at site A and many sites at site B, and then you'd have a mean number of insects at site A and a mean number of insects at site B. 3, we're investigating into the effect of light intensity on the rate of photosynthesis, so changing one variable to find out the effect on another. So this would be A, Spearman's rank correlation coefficient. And finally, if you're investigating the effectiveness of 2 antibacterial products on E. coli, then you would have lots of repeated measurements for each and a mean, and so you would be using the student t-test. Okay, so those are the answers there. So the examiner likes to ask you what stats test you would pick. So that's something we can be quite confident about coming up on the examinations, that you would need to be able to identify which stats test. And then they always want you to say why. Okay, so for the ones that you've picked correlation coefficient, it's because you were looking for a relationship or correlation between two variables. For those where you've picked the student t-test, okay, for those two, for 2 and 4, in both cases we were looking to see if there was a significant difference between two means, and they're your reasons why. Okay, so we're going to have a look at the t-test today. So as I said, stats tests are used to determine the probability of results being down to chance. So we use a t-test when you're investigating the difference between two means, and you want to know if the difference is significant or not. So you always start by writing a null hypothesis, and the null hypothesis always states that there is no significant difference between the two means. You do need to make sure that in the exam, when you write your null hypothesis, because the examiner does ask you to write a null hypothesis, it must be related to the actual practical. So if we go back to this last example here, investigation into the effectiveness of 2 antibacterial products on E. coli, if I was asked to write a null hypothesis for that experiment, I would say there is no significant difference in the mean zone of inhibition for antibacterial agent A and B. You know, so it'd have some detail of the practical within the null hypothesis. But you can learn that one on your keynotes in your revision, because then you can just adapt it by putting in a bit of the detail from the practical. Okay, so if it is found to be significant at a probability level of 0.05, so 5%, okay, then you can reject that null hypothesis because you are happy, you're certain that there is a significant difference between your two means. And another way of saying that there is a significant difference is you can say that the probability that any difference being due to chance is less than 5%. Okay, so if it's less than 5%, we're saying it's really small. So the probability that the difference was just because of chance is really small. So we're saying we're confident that it's a biological reason, a real significant difference between the two means, not just a chance difference. Okay, so how does a t-test look? Okay, pretty complicated, so I don't want you to worry too much. It looks complicated at first, okay, but although the formula itself looks a bit scary, by the time you've worked through our worked examples, you'll realise it's not that tough. We do it a step at a time in that formula. Okay, so this is the formula we use to calculate our t-value. Our t-value will allow us to work out if there is a significant difference between the two means, and is it significant, that difference, or not? Okay, once we've calculated a t-value, what we need to do then is, working at this probability level of 0.05, we need to work out a critical value. Okay, and the way you work out your critical value is you use one of these tables. Okay, a table of critical values for t, and you need to look to the right row within the table, and you find the right row by using this at the bottom here. So the number of degrees of freedom. Okay, so we need to know what degrees of freedom we're working in this particular example. So this is the number of individuals in the first sample. So in your first mean, how many different measurements were used to calculate that mean, plus the number in the second, minus 2. Okay, so then say my degrees of freedom was 6, I can then look up my critical value in the table here. And then I compare my t-value to that critical value. And from that comparison, I can work out whether the difference between the two means is significant or not. So I'm going to show you. So this worked example is in your student notes for this topic, this 15A, okay, booklet. So we've investigated the bacteria, okay, growing in the same medium, so both growing in glucose, and we want to know whether one bacteria grows more biomass in the same time period than the other. Okay, so we've repeated the experiment for each bacteria 4 times, and we've calculated a mean. Okay, so the null hypothesis we will put together for this one, remember we can use our generic one, there is no significant difference between the two means, but we do need to add the detail. So for example, in this case, there will be no significant difference between the mean mass of bacteria in culture A and B. So you can see I've just added a bit of detail related to the practical. Okay, so standard deviation's already been calculated for you. Okay, that's the same all the way through the booklet. Okay, so we're not going to try and calculate standard deviation today, but with that standard deviation, I can then do my standard deviation squared and my standard deviation squared divided by the number. So then remember, the number is the number of measurements in that sample. Okay, so we did 4 measurements, so I've divided by 4. Okay, so now we can put that into the formula. Okay, so you can see that we've just placed that data into the formula and then applied the formula through until we've calculated our t-value. Okay, so on top we've put our mean for bacteria culture A, and we've taken away our mean for culture B, and then underneath that we've kept our square root there intact, and we've done what's within the brackets first. We'd already taken the standard deviation and squared it and divided it by the number in that sample, so we've already done that, so we'll pop that in there and we add that to the same again on the other side for the second sample for bacteria B. Okay, and then we've just applied those bits in the brackets, then finally done the square root of our 312.5 and then divided, and we've got 12.99 as our t-value. So this is our calculated statistical t-value. We need to compare that to the critical value. Okay, so because there were 4 investigations, so 4 repeats for each bacteria, that was 4 in sample 1, 4 in sample 2, add them together gives me 8, take 2 away, and I'm looking at degrees of freedom 6. So this is my critical value. So that is dependent on how many repeats are done in the experiment. So it's not always the same. So you need to always calculate your degrees of freedom first and then look to the right place in the table. Okay, so 2.48 is our critical value. Okay, so when you're interpreting this, it's simple. And I always think about it as a little high jumper, and the pole is the critical value. So in this case, 2.48 is the critical value. Okay, so my calculated value, I'm comparing it. Does it go over the critical value or does it fall under it? If it goes over it, so if my calculated value for t is greater than the critical value, that means I can reject the null hypothesis. There is a significant difference between the two means. And the probability of the difference between the two means being due to chance is less than 5%. And there's a shorthand way sometimes the examiner writes that, where they write less than, or p is less than 0.05. Okay, so that would be our conclusion in this case. But imagine our calculated value did fall below that critical value, then we just actually say the complete converse. So the calculated value of t is lower than the critical value. Therefore, we'd accept the null hypothesis. There is not a significant difference between the two means. The probability of the difference being due to chance is greater than 5%, and we can write that shorthand as greater than 0.05. So p greater than 0.05. And sometimes the examiner does use that. One of the things I encourage my students to do is to just have a revision card with simply this written on. And then they can learn the outcome that they write depending on whether the calculated value is greater or lower than the critical value. By putting the shorthand in, that really helps us as well, because sometimes the examiner doesn't give you the critical value. They actually shortcut and say that the scientists concluded there was a less than 0.05 probability. And that just simply means that everything in that box there you would state. So if the examiner writes that, they mean the probability of the difference between the two means being due to chance is less than 5%. Okay, so there you go. Our calculated value is bigger than the critical value. So there's less than a 5% probability the difference in the mean mass of bacterium A and bacteria B is due to chance. We reject our null hypothesis. Okay, so you've got another worked example in your booklet. Okay, and then I would like you to stop the video now and complete questions 1 and 2 in your t-test booklet 2. Okay, so now I'm going to go through questions 1 and 2 in the booklet as well. Okay, so for question 1, we needed a null hypothesis. So you needed to make it sort of relate to the test. So there is no significant difference between the amount of hemoglobin in tests with the new kit compared to the old kit. Something like that. For my mean, I got 15.6. It doesn't matter if you've rounded to more decimal places or not. And I got a 14.4 for my mean for the old kit. Okay, and there were 6 samples in each, so I have n of 6. Standard deviation is already there for me, so I've squared it and divided it by 6, and these are my values here. I've gone to 3 decimal places, but you may not have. Okay, then I've used that in my formula, so I've put my means on top, so my first mean, take away my second mean, and then I've got my first standard deviation divided by number of samples, so sorry, standard deviation squared divided by number of samples, which is this value. And then at the bottom over here, I've got for my second sample the standard deviation squared divided by the number of samples. is this number here, 0.498. So that means I had 1.2 on top divided by the square root of 0.925. So I did the square root of 0.925, so I had 0.96. And that meant my calculated value came out at 1.25. Okay, so then going back 6 meant that comparing my table, now I was dropping down in terms of my critical value, because I had 6 values. 6, or was it going up? Sorry, 6 and 6. So that gave me 12. Take away 2, so I'm moving to 2.18 as a critical value to compare with. Okay, so my calculated value then was less than the critical value. That means this time I'm going to say there's more than a 5% probability that the differences in the means are due to chance. So I'm going to accept the null hypothesis. There's no significant difference. Okay. So for the second example then, null hypothesis, again, there is no significant difference between the mean time taken for one cell cycle between shoot and root cells in grass. So it doesn't matter exactly how you've worded it, as long as you've got no significant difference between the means and something related to the practical. Okay, so my mean, 3.1 for the shoot, 9 samples. 2.5 for the root, also 9 samples. Standard deviation's done for me, so I've squared it and divided it by 9 in each case. Okay, so then I've got my mean for my first sample, which was my shoot, my mean for my root, take away from each other, so 0.6. Then I had my standard deviation squared and divided by the number of samples for my shoot, standard deviation squared and divided by number of samples for my root. Okay, added those together, square root of those, and then I've cut it off. I think it's 2. Let's have a look. Yes, 2. Okay, so degrees of freedom this time. So it was 9 and 9. Okay, so 9 and 9 minus 2 gave me 16. I used the table, and if I go back to my table, at 16 it was 2.12 as the critical value. Okay, so then you have a bit more freedom here, so you might not have written exactly the same thing. So our calculated value of t is 2, which is below the critical value 2.12. So there's not a significant difference between the two means. Okay, and then I've shorthanded it. I've said there's greater than a 5% probability that the difference is due to chance. I accept the null hypothesis. Okay, so remember, stats is always on the exam. Okay, so we need to make sure that we've done a lot of practice before the exam. Okay, but you won't have to calculate t-values outside of your lab book. Okay, so all of this prep work in your your t-test booklet, the calculation bits are for practice for your lab book, so you'll be able to apply that in your practical work. But the interpreting the results, that is on your exam, so that we will need to be able to write these paragraphs, we'll be able to have to interpret these values. Okay, so just a little bit more practice. Can you choose the right test? Okay, so I've got 3 examples here. You can go back in the PowerPoint and look at that flow chart again if you need to. Of our 3 tests, which would you use for each of these experiments? Okay, stop and have a go and then join me back. Okay, so 1 If you wanted to find out if your data, what stats test would you use if your data was categoric? So in other words, falls into categories like pink and white flowers. Okay, well, if it falls into categories, what you'd be counting up is a frequency. How many of the flowers are white and how many of the flowers are pink. So whenever it's categoric data, it's going to be chi-squared. Okay, what about 2? Which statistical test will you use if you want to compare the 2 mean values to see if they're significantly different or not? I'm hoping you've got student t-test there. And finally, which stats test would you use to see if there is a significant correlation between 2 variables? So correlation coefficient, or you can call it Spearman's rank. Okay, so there are your 3 tests. And you do need to know why you use them in each case. So for chi-squared, as well as talking about investigating and counting up frequencies, you could use that word that it's categoric data. That's another way of saying that in your explanations. Okay, so sometimes in the exam, they just ask you to do something like this. Write a paragraph using the terms probability and chance if the difference or association from the stats results at p 0.05 has a calculated value greater than the critical value. So stop and have a go. Can you do it from memory now? Right, so if it is greater than the critical value, you should be saying everything in this top box. Reject your null hypothesis. There is a significant difference between the 2 means, or between there is a significant association between the 2 variables if it's an association. The probability of the difference or association being due to chance is less than 5%. That's what you should be saying. Okay, so this is a recent exam question. So here is the mark scheme. Look, the probability of the difference or association being due to chance is less than 5%. Therefore, there is a significant difference or association between the 2 means or variables. Now, if there is an experiment you are given, then you need to name them and link back to the investigation here. And you reject the null hypothesis. So you can literally make a beautiful little flashcard with all of that on and learn it off by heart to give your concluding paragraphs for your stats. They're always the same, we're just changing one word or so. Okay, now, then the examiner has a bit of fun, okay? What if the examiner says something mean to you like this? If the difference between the 2 means or the association between 2 variables is less than 0.001 in terms of probability, what would you say that means? Remember, you can shorthand. So Dave's shorthanded, but we're used to seeing the shorthand less than p 0.05, which we know means that there's a less than 5% probability that the difference or association is due to chance. So what about this then? What does this mean? Well, the exam board have just gone to a higher level of probability. So the probability now, the difference or association being due to chance, is even smaller. So it's even more significantly different, or a significant relationship, because the probability of it being due to chance is tiny. It's less than 0.1%, which is the kind of level that you'd want to be using if you were doing paternity tests. So this is the probability level we do use when we do DNA testing. Okay, that means there is a significant difference or association, but it's even more significant if the p is smaller. Okay, so the examiner would still ask you to give the same, reject the null hypothesis. The only thing that's changed in that information is instead of 5%, it's 0.1%. Tricksy, but we'll be ready for them. We'll practice. Okay, so this is an example of a recent exam question where they used these probability values. Okay, and they said they were looking at the mean number of butterflies in the canopy and the understory. Okay, and they were asking whether there was a significant difference between the number in the canopy and the understory. Okay, but if you look at the results, it was significantly different for some, but not for others. And because of the value, we could say what level of probability we were working to. So all the ones in yellow, we were able to say the probability of this difference being due to chance was less than 0.1. Therefore, there was a significant difference for each of these species between the mean in the canopy and the mean in the understory. And we would reject the null hypothesis that there was no significant difference in the mean number of butterflies in those two areas for those 5 different species. Okay, now in the exam, the students actually had to name these species and to give that result for all 5 of them. And then they got an extra set of marks for identifying for this one species that because we were greater than 5%, the probability of the difference between the mean number of butterflies in the canopy and the understory for this species was greater than 5%. Okay, so the probability of the difference being due to chance was greater than 5%. So therefore it was not significantly different between those two. Okay, and in this case, she says there's an error there, would accept. We would accept the null hypothesis that there is no significant difference. Okay, right, so what I've done is I've set up some extra t-test and stats tasks for you to do. Okay, now I know that this is one of those things that sometimes takes people a bit of practice and having to go on their own. So I've done that extra one for you, and this week on Thursday in the tutorial, that's what I'm going to go through, those t-test extra tasks, and answer any questions on the t-test and the statistics. Okay, and I'll have some extra examples, some extra exam questions to take away from that tutorial. If you can't come to the Thursday tutorial, that's fine. We will send out a mark scheme to this additional task, these extra tasks. But obviously if you can make it, then that's where you can get your answers to that, as well as explanations. Okay, right, so crack on with those extra tasks, and then we've got one more video this week to view as well.